Okay, so good evening, everybody. It's great to see so many friends and colleagues here this evening and family, all very, very welcome. My name is Mike Hanna. It's my privilege to be the director of the Institute of Neurology. This is really an amazing institution with great colleagues dedicated to deeply understanding how the brain works and finding treatments for often devastating diseases, as well as educating the next generation. And tonight we're going to celebrate a major milestone for two of our really great colleagues, Rob and Rimona. Becoming a professor at UCL is a huge achievement. It's really very difficult to be a professor here. It's actually much harder than at any other university, but I'm obviously a little bit biased. So, as is tradition, I'm going to introduce both of these lecturers, and then there'll be a vote of thanks after each. I will give Rob's vote of thanks, and I'm very pleased to welcome Geraint Rees, the Vice Provost for Research, who's going to give the vote of thanks for Rimona. So that's great. Thank you for coming, Geraint. So turning first to Rob. Rob Pitceathly is now Professor of Clinical Neurology and Mitochondrial Medicine here at Queen's Square. He's an honorary consultant neurologist in the hospital. He completed his preclinical training in clinical medicine and clinical medical degree at St Andrews and Manchester University before commencing postgraduate specialist neurology training first in Manchester, and then he subsequently came to London, where he took a PhD with us in mitochondrial disease before obtaining an NIHR academic clinical lecturer post in London. And then in March 2019, he secured a prestigious MRC Clinician Scientist Fellowship and became an honorary consultant in the hospital and established his research group. He also is a very active clinician and co-leads an NHS England nationally commissioned service for rare mitochondrial disorders, which serves patients throughout the country. And just recently, Rob became head of the research department of neuromuscular disease here at the Institute. As I'm sure we're going to hear, Rob's research combines clinical observation with laboratory approaches in the best traditions of Queen Square to deeply understand the molecular basis and pathophysiological mechanisms of primary respiratory mitochondrial diseases, with the aim ultimately of trying to develop treatments. He's really extensively published, he has a national and international reputation, and as I say, has secured major research funding. So I'm really delighted to welcome Rob to give his inaugural lecture.
Thank you, Mike, for the kind words. It's a huge pleasure for me to be here today to celebrate my professorship with all of you and those online. I've learned all sorts of important lessons during my academic career, but some stand out because they've been a constant feature at each stage in the journey. And these can really be summed up in 3 words: teamwork, persistence, and family. These are recurring themes in the talk, during which I hope to convince you that I reached this point not through some exceptional talent, but rather through more ordinary attributes of resilience, hard work and focus, with a clear set of objectives and the support of a fantastic group of colleagues, friends, and an amazing and incredibly tolerant family. I've entitled my talk Fuel, Fault, Fix, a journey in mitochondrial translation, but before I talk about my personal background and research journey, I felt the imperative need to begin with a question which I'm asked on an almost daily basis by patients and colleagues alike, many of whom have known me for several years. And that question is, how do I pronounce my surname? So there are several iterations, mine's highlighted there, probably all with the same ancestral origin. I pronounce it Pitceathly. However, my brother, who's actually here today, Oliver, pronounces it
Pitceathly.
So even within our family, we don't agree. So when in doubt, what do we now do? We ask ChatGPT. So ChatGPT tells us that the name is of Scottish origin, which we already knew. In fact, my brother recently travelled to Scotland to our ancestral home, Pitceathly, Wales, and took those photos in Perthshire. There are lots of variants, it says, all with the same pronunciation, so this is looking good. And the name should be typically pronounced phonetically as pit as in pit, key as in sea, and thly as in flea, so Pitceathly. And then it directs you to an external website where it gives you an audio.
Pitceathly.
So it didn't help at all. So the phonetic odyssey continues. So I suppose my journey begins with the town in which I was born, and that was Stockport in Greater Manchester. I'm sure it needs no introduction, often referred to as a jewel of the north. It's nestled 4 miles to the south of Manchester city centre and to the west of the Pennines, and it's the origin of the River Mersey, at the confluence of the River Tame and the River Goyt, shown in this picturesque photo here. It was a central hub for the hatting and later the silk industry, and the town expanded rapidly during the Industrial Revolution, although economic growth did take its toll. And in the 19th century, philosopher Friedrich Engels wrote that Stockport was renowned as one of the duskiest, smokiest holes in the whole of the industrial area. Well-known landmarks in Stockport include its viaduct, which you can see in these 2 photos here, with Lawrence Lowry just standing in the foreground of the top right photo. And it used 11 million bricks to construct, and is considered one of the world's largest brick structures. And there's the Hat Museum. Famous Stockfordians include Fred Perry, winner of 8 tennis Grand Slam titles, and the Queen, or at least Claire Foy, who played the Queen in the eponymous historical drama series. Finally, Stockport is of course home to the mighty Stockport County football team. I grew up in a family of non-medics, but inherited a determination and single-mindedness I think both my parents exhibited through their occupations and outside interests. My mum was a social worker and later trained as a psychotherapist, specialising in bereavement counselling at the Christie Cancer Hospital in Manchester. However, she was also a talented runner with a personal best for running the 10K of 39 minutes 42 seconds. And those that run the annual CNMD 10K with me will attest that her running abilities definitely skipped a generation. My dad was a piano teacher attending the Royal Northern College of Music in Manchester and was later head of the keyboard department at the prestigious Manchester Chetham School of Music. And he combined these musical talents with perhaps an incongruous passion for powerlifting, winning multiple British championships and achieving 3rd place in the 1972 World Championship in Harrisburg, Pennsylvania, where he actually held the world record for bench press in the 110 kilogram category for a total of 20 minutes. My brother inherited some of these creative traits and attended Chelsea College of Arts. So I managed to scrape together the grades needed to study medicine at the University of St Andrews in Scotland. And St Andrews is actually the oldest university in Scotland, 3rd oldest in the English-speaking world. And the town sits in an incredible place on the east coast in Fife, amongst some stunning scenery, and is of course the home of golf in the Old Course, the most frequent venue of the Open Championship. It has a number of long-standing traditions, including the Sunday morning pier walk, where the students wear this customary red gowns, and also Raisin Weekend, which is where first-year student children thank their academic parents for welcoming them to the town through the medium of foam. Of course, Will and Kate attended St Andrews University, although after I'd left. Given I was from Stockport, and the central picture there is the medical school, the Butte building. Given I was from Stockport, I think university accommodation officers thought it appropriate to allocate me what was arguably the finest hall of residence the university had to offer at the time. Not Hamilton Hall, which was situated overlooking the Old Course, but rather David Russell Hall, further out from town. I was always surprised it didn't feature more heavily in the university prospectus. I moved back south to Manchester for my clinical medical training, which was obviously a very different student experience, but one that I thoroughly enjoyed. It gave me the opportunity to enjoy the city as a student rather than a resident. Although the city has changed enormously since from my time as a medical student, I've included some nostalgic photos here that those of you who know the city may recognise. My wife Debbie comes from a long line of very hardcore Manchester City supporters, but she very graciously still allowed me to include a photo of Old Trafford, which is just there on the left-hand side. I spent most of my clinical medical training at Wythenshawe Hospital in South Manchester, which holds a very special place in my heart, as it's where I met Debbie during a week of night shifts. After qualifying in 2003, I remained in Manchester for my house officer and SHO rotations, spending time at the Christie Hospital, North Manchester General Hospital and Wythenshawe Hospitals. Some of you may remember the disastrous medical training application service that was adopted during modernising medical careers in 2007. And I was actually offered a training number in haematology in London, which I subsequently gave up after falling in love with neurology during a 6-month SHO post, and instead took up a locum neurology SPR post at Salford Royal Hospital. This was one of the best decisions I ever made and was the start of an uncompromising streak which has perhaps been a feature of my career. And it was during this post that I met Mark Roberts, consultant neurologist and myologist, a proud Welshman and one of the best clinical neurologists I've ever had the pleasure of working for. He inspired me to pursue a career in muscle diseases and was instrumental in my application for a PhD in mitochondrial diseases after a conversation with Mike at a myology conference where it was mentioned there was potentially a mitochondrial clinical fellow position at the MRC Centre for Neuromuscular Diseases. So I joined the MRC Centre in 2009 as a clinical research fellow, completing my PhD in the molecular and clinical aspects of mitochondrial diseases in 2013, under the supervision of Mike and Shamima Rahman, whose training in clinical and fundamental aspects of mitochondrial disease and biology really influenced and shaped my subsequent postdoctoral academic career. I also received training from two very inspirational figures, both of whom had a major impact on me during my PhD. The first was Mary Sweeney. Mary was a clinical scientist and head of neurogenetics, taking over from Mary Davis. She had huge expertise and a passion for mitochondrial disease diagnostics and spent many a painful hour, for her anyway, on the 6th floor of Queen Square House, trying to teach me genetics, how to design primers and how to perform Sanger sequencing and restriction fragment length polymorphism. She sadly died very prematurely, but she enhanced the lives of those who knew her immeasurably, and I was incredibly fortunate to have had those hours with her in the lab. The second person was Jan Willem Tarman. Jan Willem is based at the Royal Free Hospital, where he continues to run a mitochondrial research laboratory. He's a biochemist and fundamental mitochondrial scientist in the purest sense of the word. He trained me in several techniques that were pivotal to my research, and he continues to train members from my lab to this day. During my time at the Centre for Neuromuscular Diseases, I also benefited hugely from exposure to outstanding leadership and mentorship gained from Mike, Mary and Doug Turnbull in Newcastle. These interactions significantly shaped my own career and the choices that I subsequently made. And of course, I have to pay special tribute to my amazing CNMD colleagues whose commitment to the clinical care and translational research in neuromuscular diseases is unrivaled. These photos really underline the importance of being surrounded by an amazing team, a recurring and enabling theme in my career. After my PhD, I returned to clinical neurology training, working at St Thomas's, King's College Hospital and National Hospital as an academic clinical lecturer. And as my CCT date approached, I began applying for clinician scientist fellowships. I think it's really important to promote success, but I also think we should celebrate the failures that often underpin that success. And this is my journey towards securing a clinician scientist fellowship between June 2016 and 2018. And I think this really emphasises the importance of persistence. If at first you don't succeed, try and keep trying repeatedly. I applied at least 10 times with 2 interviews before I managed to secure my MRC Clinician Scientist Fellowship. In doing so, I returned to the now Department of Neuromuscular Diseases in 2018 to set up my lab. And at this stage, I should really thank Linda Greensmith for all her support in this endeavour, but also Nicole Falabella, a senior postdoc in my lab, who agreed to join it when it looked like the photo on the left, transforming it to the photo on the right. While establishing the lab at IoN, I took over as co-lead for the mitochondrial highly specialised service with Mike, and this service really comprises a fantastic interdisciplinary group providing clinical and diagnostic care for all patients with mitochondrial disease, with partner sites in Oxford and Newcastle, so we provide a national service. And the clinic is intrinsically linked to all of my research, both the discovery and the translational. And these photos really are just to emphasise the size of the team and their different backgrounds, clinical, admin and diagnostic teams that all provide fantastic care for our patients. So, I've talked about mitochondria a little bit, but what are they? So, this is how they're often depicted in textbooks. This is obviously not how they really appear, but it's a useful diagram to use. So, they're double membrane bound organelles. The inner mitochondrial membrane is highly convoluted, providing a large surface area for all the very important metabolic reactions that take place within them. But actually, this is what they truly look like. They're incredibly dynamic, constantly fusing and dividing, exchanging information about the homeostatic state and metabolism of the cell, allowing them to regulate themselves according to the energy demands. And often when we're referring to mitochondrial diseases, it's this process, which is just one of many that take place within them, oxidative phosphorylation, which is the sequential transfer of electrons across a series of redox reactions taken place on these large multimeric enzyme complexes. In doing so, hydrogen are pumped from the mitochondrial matrix into the intermembrane space, thereby setting up an electrochemical gradient, which is harnessed by the terminal component of oxidative phosphorylation, ATP synthase or complex 5, which uses that to convert ATP from ADP and inorganic phosphate. Mitochondria are really interesting because there are hundreds to thousands in all the cells of the body, depending on the energy demands of that cell. But they contain their own genome, a small circle of DNA, around 16,500 base pairs, that encodes 37 genes, 13 of which are subunits of those enzymes that are producing energy, the respiratory chain. The remaining 24 are involved in intramitochondrial protein synthesis of those 13 polypeptide subunits. And being DNA, mitochondrial DNA can mutate, and the first mutation within mitochondrial DNA associated with human disease was identified right here at the Institute of Neurology with Ian Holt, who's sitting in the audience today, along with Anita Harding and John Morgan Hughes. And what they did for the first time was look at mitochondrial DNA that had been extracted from muscle tissue. And they used multiple restriction endonuclease digest analysis, where they detected 2 clonal populations of DNA in patients with chronic progressive external ophthalmoplegia and mitochondrial myopathy. And this is taken from their paper in Nature, and what this shows in patients in the muscle tissue, not the blood, 2 populations of mitochondrial DNA molecules when they use this digest and run on an agarose gel. And what that shows is there are 2 populations of mitochondrial DNA, one that's got a big deletion, a big bit that's missing. And when you linearize it, it runs further on the gel, faster on the gel. And so that second band is the abnormal deleted band. And since that time, over 250 pathogenic variants have been identified in this very small circle of DNA. But mitochondrial DNA-related diseases have a number of other interesting attributes. First of all, as you all know, mitochondria are maternally inherited, and so someone with a mother with a mitochondrial DNA mutation will pass that to all her children. The level of the mutation within the cell can vary because we have hundreds to thousands of mitochondria in every cell. You can end up with different populations of mitochondria, some with the mutation, some without the mutation, something known as heteroplasmy. And as you might expect, the higher the level of the mutant mitochondria, the more likely the cell is to fail. And there's a critical biochemical threshold that's dependent on the cell and also the mutation where that occurs. There are also a number of bottlenecks that occur within mitochondrial genetics, and one of the most important occurs in an early development of the primordial germ cell, when there's a a significant reduction in the number of mitochondria, followed by random segregation into daughter cells following expansion of those mitochondria. And what that means is you end up with eggs within a woman that has a different level of mutant mitochondria. And so the offspring can have different levels of that mutation and very different severity phenotypes as a consequence. But that's all about mitochondrial DNA, but actually there are a large number of nuclear genes that are also very important for mitochondria to function properly, probably around 1,500, and mutations in over 350 to 400 of those have also been associated with Mendelian mitochondrial disease. So you can see why mitochondrial diseases are typically thought to be incredibly heterogeneous, both clinically and also in terms of their severity, because of this dual genomic expression of mitochondrial proteins and the complexity of mitochondrial DNA-related diseases. So my research really attempts to bridge clinical observations and laboratory science to better understand the molecular mechanisms driving primary mitochondrial disease, but also mitochondrial dysfunction that occurs within other neuromuscular and neurodegenerative disorders, ultimately with the goal of developing therapies. Our fundamental science really focuses on new disease genes and disease mechanisms, but with a particular interest in those relating to cardiolipin biosynthesis. But we're also very interested in other aspects of mitochondrial biology through reverse translation. So the clinical observations, the biochemical molecular findings that arise from the clinical and diagnostic services, and then using those to inform basic aspects of mitochondrial biology. So impaired phospholipid metabolism has become of high interest over the past 5 or 10 years. And there's a novel class of primary mitochondrial diseases that relates to this due to problems with the mitochondrial membrane itself. And one phospholipid in particular, cardiolipin, is of particular interest. And it's perhaps unsurprising that this has been associated with mitochondrial disease given its critical role in mitochondrial function and structure. It's a unique phospholipid that's mainly present in the inner mitochondrial membrane, and it's got a distinctive conical shape, and that's because it's got 4 acyl side chains rather than the typical 2 canonical acyl side chains typically found in other phospholipids. And what that means is it creates a negative curvature in membranes, and that's actually why the inner mitochondrial membrane is highly convoluted, because cardiolipin is concentrated in those areas of negative curvature. What's also really interesting about cardiolipin is because it has 4 acyl chains that can be any length and any saturation, it can be incredibly complex and diverse, and this is dependent on the tissue, and it's likely that that has a regulatory role, which I'll come on to in a minute. In addition to its role in membrane architecture, cardiolipin participates in a wide range of mitochondrial processes, including formation and maintenance of protein-protein and protein-membrane interactions, stabilization of mitochondrial respiratory chain complexes, assembly of complex 5 dimers, and organization of supercomplexes. And under stress, cardiolipin is externalized on the outer mitochondrial membrane and functions as a signaling molecule to promote mitophagy and apoptotic signaling pathways. In mammalian cells, de novo synthesis of cardiolipin is divided into 2 steps. The de novo synthesis, but then remodeling of those 4 acyl chains that are specific to the tissue that it's being generated in. It's a multi-step process that takes place with multiple proteins, starting with the transport of phosphatidic acid from the endoplasmic reticulum across to the inner mitochondrial membrane, where this biosynthetic pathway takes place. And as you can imagine, each one of these proteins is encoded by a different gene, and each one of those genes can be potentially mutated to cause human disease. And actually, we've been involved in several genes in this pathway that have recently been associated with human disease since the very original, which was tafazin and Barth syndrome, which was described in the early 80s. What's interesting, aside from all the bioenergetic defects that we see associated with cardiolipin-related mitochondrial disease, is that there's some consistency with the phenotypes, particularly the involvement of muscle tissue, both skeletal muscle and cardiac tissue. And that must somehow hint towards a specificity of the cardiolipin species that is within muscle tissue, and its vulnerability to mutation within these genes. And that's something we're actively exploring in the lab. Cardiolipin has become a target for a number of treatments, and a company called Stealth Biotherapeutics has produced a drug called elamipretide, which is a tetrapeptide which actually binds to cardiolipin on the inner mitochondrial membrane, stabilizing and improving oxidative phosphorylation efficiency. And that's actually at the late stages of being approved for Barth syndrome, and has been in a number of clinical trials for other common diseases in which cardiolipin has been implicated, including heart failure. So there are a number of future directions that we're taking this work in the lab. So pathways involved in just normal cardiolipin metabolism still remain pretty poorly understood. It's likely that that cardiolipin will contribute to several other currently uncharacterized aspects of cellular homeostasis and human health. Mutations in cardiolipin-related genes are still under-recognized, and there are a number of genes that haven't been associated with human disease that we're likely to see. And actually increasing cardiolipin biosynthesis represents a therapeutic option for both mitochondrial but also other diseases as well. And we're involved in companies that are already developing those. We're also very interested in understanding other aspects of basic mitochondrial function and respiratory chain structure. And one good example of this is a gene that was called NDUFA4 when I first started working on it during my PhD. And this was a gene that was thought to encode a subunit of complex 1. But we identified patients in our clinic that actually had complex 4 deficiency. And as I say, this is a really good example of reverse translation. What was unusual about these patients is they had Lee-like syndrome, but actually a relatively mild phenotype. And so these were patients that were living to their third or fourth decade rather than dying during childhood. The explanation for the misattribution of NDUFA4 as a complex 1 subunit was thought to be relating to the harshness of the detergent used to isolate the different respiratory chain enzyme complexes, which meant the subunit got dissociated from complex 4 and ended up as a contaminant of complex 1 and explained its incorrect assignment. What was difficult to explain, though, was the fact that the patients that we were seeing with a relatively mild phenotype had splice site mutations, so loss of function mutations, with absolutely no detectable NDUFA4, now called COXFA4, protein levels, but still had residual complex 4 enzyme activity, and that was quite difficult to explain. And it took us 12 years to do this, and this is work that's really been spearheaded by Nicole in the lab. And what we've managed to do over that time is collect a number of other families with COXFA4 mutations, and work by Nicole in the lab has established the subunit is definitely a subunit of complex 4, and it's involved in the late stage of complex 4 during its assembly. Importantly, what McCall's identified is the upregulation of a very closely related protein called COXFA4L2 is compensating functionally for the absence of COXFA4 protein and effectively partially rescuing the biochemical deficiency of complex 4. And this is what explains the relatively mild adult phenotype compared with other genetic causes of Lee syndrome, despite the absence of the mutated protein. It also now represents the most common complex 4 subunit to be associated with human disease, and as a consequence, we're actively pursuing potential genetic therapy through AAV for this cohort of patients. So another important focus of the lab is molecular diagnostics and integrative multi-omics, particularly with a view of transferring these advanced technologies from the research setting into the diagnostic setting. And there's really been a paradigm shift with primary mitochondrial disease diagnostics in the past 10 years from what was quite complicated, using biochemistry, histochemistry, and genetics, using multiple different tissues to make a diagnosis, to very much a genomic-led diagnostic approach, as with all other neurogenetic conditions. However, the Holy Grail is really a single test, a non-invasive test that we can use for all patients to diagnose, because we're still using quite a large number of different techniques to reach a diagnosis. And the challenges for developing this single test are very similar when thinking about whole genome sequencing as with other rare diseases, apart from one specific point around mitochondrial DNA, and that's new mites. So these are nuclear-embedded mitochondrial DNA sequences, usually less than 500, but often less than 200 base pairs, that get integrated, these sequences being integrated into the nuclear genome, but they're essentially homologous to the mitochondrial DNA, and sometimes variants in those nuclear new mites can be mistaken for mitochondrial DNA mutations. But they can be worked out through various bioinformatic tools. And actually the challenge with this single test is more the mitochondrial DNA, despite how small it is. And the reason for that is you need to identify the variant, and you need to be able to quantify the variant and its variant allele fraction. And you need to do that very accurately, because even low-level mutations can be relevant. And so you need extremely deep coverage in whatever technology you're using to analyze that. The other challenge with mitochondrial DNA when developing a single test is its temporal shift in heteroplasmy. So in dividing tissues such as blood, we see a drop in the mitochondrial DNA mutant level, and that's because there's a selection pressure against the dividing tissues. But actually, in post-mitotic tissues, such as muscle tissue, you can sometimes see clonal expansion of the mutation. And consequently, we often have to sequence multiple tissues to make a diagnosis. And that can be a challenge, particularly in terms of the quantity and quality of the DNA that you extract, for instance, when using muscle tissue, using manual homogenization techniques due to fatty infiltration and freeze thaw. So all of these need to be considered when developing this single test. But this is a situation that we're trying to move on from, this time-consuming, expensive, multi-step process in reaching a molecular diagnosis of mitochondrial disease. And this just is for mitochondrial DNA. And so we're trying to do this through long-read sequencing. We think this is going to be the solution to looking at mitochondrial DNA and nuclear DNA in parallel and making a diagnosis as quickly as possible. And so we've been looking at Revio hi-fi data. So this is long-read sequencing where you're actually sequencing tens of thousands of base pairs, and so you can actually sequence the whole of the mitochondrial DNA molecule in a single read. And that obviously has potential for improved analysis of structural variants, so these big deletions that are thousands of base pairs and new mites as well. It's highly accurate because of the technology, the circular consensus sequencing that's being used, and can reliably detect very low variant allele frequency, so low heteroplasmy levels. And finally, with a new chemistry that's being developed, we're allowed to sequence with much lower DNA input than previously when it first came to market. And that's obviously a huge advantage when looking at DNA, particularly when extracted from muscle tissue. Finally, the group is also involved with several aspects of translational research, including drug development, both in vitro and in vivo, small molecule testing, the development of genetic therapies, and outcome measures and clinical trials for which there's increasing interest for mitochondrial diseases. And many of these endeavours really link to an important centre that's recently been funded by LifeArt, the LifeArt Centre to treat mitochondrial disorders, which is being led by Patrick Chinnery from Cambridge, but also by Mike at UCL and Rob Taylor in Newcastle. And this centre really aims to tackle three big challenges within mitochondrial diseases, and that's diagnostics, biomarkers, and new treatments, and that really synergises very well with the interest within our lab. And the aim is for that to be within the next 5 years. And one reason for this initiative is, despite this huge interest in developing treatments for mitochondrial diseases, and there are a lot of targets, as you might anticipate, for mitochondrial diseases, because the number of pathways you can target within mitochondria, is we still don't have a therapy for the majority of mitochondrial diseases, despite this exponential increase in clinical trials. And there are really two major reasons for that. The first is that the outcome measures that we have for mitochondrial diseases haven't kept pace with a number of clinical trials. The second is very few clinically meaningful preclinical animal models, predictive animal models of disease, to test the efficacy of these treatments. So often what we're seeing is these treatments are going from in vitro studies with some safety in rat and mouse models, but then they're going straight into clinical studies. And this is the size of the problem. So this was just a consensus study of experts looking at mitochondrial myopathy, and they came up with this list of outcome measures. And when you see a list of outcome measures that's this long, you know that they've not agreed, and there isn't a one-size-fits-all. And this is just for primary mitochondrial myopathy. And just consider the fact that these patients are often relatively disabled by fatigue, and so they can't undertake many of these physical outcome measures in one sitting. And so what we're doing is we're looking at imaging outcome measures, given the history of this at Queen Square across neuromuscular diseases and the expertise that we have. And this is being led by Dr. Chiara Pitceathly, who's using muscle MRI in primary mitochondrial myopathy across three different techniques. The first being quantitative MRI, the second 31-phosphorus magnetic resonance spectroscopy, and the third oxygen extraction fraction, which is a sort of novel technique in muscle anyway, using MRI. And this is just some of the data that Chiara has generated. The first was a longitudinal study over 12 months at baseline and 12 months, and what she showed using quantitative MRI is that fat fraction within muscle tissue of patients with mitochondrial disease, specifically chronic progressive external ophthalmoplegia and MELAS, increased by 0.3%. And although that doesn't sound like a lot, that was statistically significant when compared with controls, and is therefore sensitive enough to use during the lifetime of a clinical trial, unlike the patient-reported outcome measures and physical outcome measures that she also looked at within the lifetime of the study, which didn't change over that 12-month period. The second technique is 31-phosphorus spectroscopy. And this is really this technique really enables measurement of the rate of recovery of high-energy phosphate-containing compounds, and in our case, what we're interested in is ATP before and after exercise. And what you can see here is the recovery period for the patients with CPO and 3243 is much longer than healthy controls. And I think arguably this is something that is going to be particularly useful and sensitive for clinical studies with molecules that are targeting the efficiency of oxidative phosphorylation. And finally, muscle oxygen extraction fraction. So this is an innovative technique that measures the balance between oxygen delivery, so blood flow, and oxygen consumption, so metabolism via mitochondria. And this has potential use actually in all sorts of different genetic and acquired muscle diseases that impair oxidative phosphorylation. But again, we're looking in mitochondrial myopathy, and we're in the setup phase of that. But that essentially involves some quite complicated activity that within the scanner. So a cuff is put around the thigh, it's compressed with 20 millimeters of mercury above the systolic over a 5-minute period, and then measurements are taken before, during, and after that. The second issue that I mentioned is the problem that we have, particularly with primary mitochondrial diseases and predictive mouse models of disease. And we're trying to address this through a major initiative that the MRC funded, a £22 million funding initiative to create a network across the UK of disease-focused clusters with the Maryline Centre, which is a core MRC facility in Harwell, being the hub for that network to do the animal phenotyping. And this was really to advance disease phenotyping, but also to set up predictive models of disease to facilitate preclinical testing of small molecules and genetic therapies to accelerate read-through of drugs into the clinic. And so we secured a mitochondria cluster, or mitocluster, and this involves colleagues, interdisciplinary colleagues across England with the Maryline Science Centre at its core. And the main aim of the cluster is to develop a phenotyping platform for mitochondrial dysfunction, so not just for mitochondrial diseases, but for any disease in which you want to explore the possibility of mitochondrial dysfunction and be a contributor at the Mary Lyon Centre, who do all the live phenotyping, and then any specialist ex vivo experiments are done in one of the MitoCluster partner sites. We're also assembling a gold standard UK platform for editing the mitochondrial genome, now that there are base editors that allow us to manipulate mitochondrial DNA, introduce disease-relevant mutations within mitochondrial DNA to understand them better, but also to correct them, whereas in the past we've not been able to do that. We're trying to develop a platform where we can actually deliver that efficiently and offer that to the UK academic community. We're delivering training courses and also partnering with industry who are actively testing their small molecules in our mouse models. And so what we've done is we've taken the existing pipeline at the Mary Lyon Centre, which is already incredibly specialised and broad, and we've cherry-picked those that are particularly relevant for mitochondrial diseases into our pipeline, and we've started phenotyping existing mouse models of mitochondrial disease to see if our pipeline is better than the standard and can pick up phenotypes that perhaps were overlooked in the past. We've also adapted that pipeline to introduce very specific challenges that are relevant to mitochondria, so metabolic challenges, including environmental challenges, dietary challenges, immune-based challenges, behavioural challenges, and also additional protocols. So, for instance, having an ageing protocol to make sure that we pick up age-related phenotypes, and also drugs that can affect the ability of the mitochondrial DNA to replicate, to actually exaggerate phenotypes that perhaps would have been dormant without these challenges. And finally, we've adapted the pipeline to enable us to do preclinical testing specifically of drugs that are particular assets that companies are interested in, but also genetic therapies as well. So my final slide before my acknowledgements reflects the most recent development in my academic career, which is the role I've taken on as head of the research department for the Department of Neuromuscular Diseases. And one major motivator for applying for this role was that over the past few years I've really moved from receiving satisfaction from my own achievements to being motivated by seeing others succeed. And this position really provides a unique platform to support colleagues do that, achieve their goals, whatever they may be, while building on the success of the department as a global leader in discovery and translational neuromuscular disease research. And it's a huge honour for me, particularly as it coincides with a pivotal time during the Institute's history with the imminent move to Gray's Inn Road and a groundbreaking era in neuromuscular disease translational research. So I'm incredibly excited to take it on. So arguably the most stressful slide of the presentation is this one. I've tried to thank everyone or include a photo as I've gone along, but I want to pay particular thanks to everyone in my lab, the London Highly Specialised Service for Rare Mitochondrial Disorders, the Queen Square Centre for Neuromuscular Diseases and the Department of Neuromuscular Diseases, collaborators, mentors, students and funders, and of course all the patients who are at the very centre of everything we do across the Institute. Finally, how else could I end but with an ode to my family, the importance of which is the third and most important lesson I've learned over the past 2 decades and has enabled me to reach this point in my career. Without their amazing support, particularly my 3 children, Theo, Dahlia and Saul, and Debbie, an incredibly successful geriatrician whose husband I'm lucky to be able to call myself, I wouldn't have been afforded the time, space, or belief to achieve what I have. So to them, I say thank you from the bottom of my heart. Thank you.
Great. So, Derek, do not put up my next slide yet. Okay. So, I want to thank Rob for a really excellent lecture, which I think demonstrates many of Rob's very genuine qualities and attributes. Rob is really an excellent molecular scientist, as I think is evident from his lecture. He's also a really good clinical neurologist, and is really dedicated to his patients and to finding a treatment for mitochondrial disease. And he's a great advocate for patients, working with the charities. Rob is a humble man, as you experienced there, but I have to say he has a steely determination like I have never seen before, actually. He's extremely determined, which I think is evident from his never giving up approach to getting funding, which I think is a good lesson. And Rob, above all, is a team player. He was a great team player as part of a team when he first came, and now has developed a really high-functioning, diverse, very successful team himself, which I think is a great credit to Rob. So I just want to show one data slide. I'm going to swap over the slides. So this data is so recent, I didn't have time to do a fancy slide. I just had to write this out myself because it's data that Rob hasn't actually seen. So this is a graph of a surrogate marker of human muscle mitochondrial ATP production. Now, as is the case with all BEST experiments, this is actually an experiment that includes Rob's muscle. And Rob's, the surrogate marker is the time it takes to run 10 kilometers, which is totally dependent on ATP production from your mitochondria. So the top line is Rob's dramatic improvement over time, along with his increasing publications, his increasing H-index, his grants. His time to run 10 kilometers has progressively fallen over the last 10 years, from when he first arrived and there was about 3 people doing it to the big team that does it now. Sadly, the lower line is my graph. So I used to be moderately good, but it's getting worse and worse. And actually, this year, which is in 3 weeks, I think that it may be a pivotal inflection when Rob may actually be ahead. And actually, this is how Rob looks at the beginning of 10K, but it's different at the end. But he's definitely getting fitter and fitter. And of course, he's part of a fantastic team who have done it every year for the last 15 years. So I think we should all just thank Rob for a fantastic lecture. Thanks. So I'm really, really pleased to introduce the second lecturer. So Rimona is now Professor of Neurology at the UCL Dementia Research Centre and an Honorary Consultant Neurologist at the National Hospital. She holds an extremely prestigious Wellcome Career Development Award to investigate dementia in Parkinson's using imaging and plasma biomarkers. Alongside this, as many of you will know, she's set up and leads an excellent clinical service managing patients with Parkinson's dementia and dementia with Lewy bodies. She has a really extensive, impressive publication record and is highly cited and is a real international expert now in her field. Rimona studied medicine at Downing College, Cambridge, followed by UCL. Her PhD examined the integration of visual signals in the healthy and damaged brain, and she was awarded a postdoctoral UCL Excellence Fellowship to study visual changes in Parkinson's. Her ultimate aim is to develop treatments to slow the progression of dementia in Parkinson's and dementia with Lewy bodies. So I'm really delighted to invite Rimona to give her lecture.
Wow, thank you for coming. It's really amazing to have, and slightly strange, to have a combination of family, friends, colleagues, and colleagues who've come from around the country. Wow. Amazing, cutting high people. So thank you for being here. Right, so I'm going to start with the beginning, otherwise I'm going to get too emotional. So this is where I began. This is my parents, Jonathan and Judith, holding me outside the Middlesex Hospital in the 1970s. You might be able to tell from their clothes. I hope that I've learned from the values that my parents have taught me. So my dad trained as an engineer. He spent his childhood taking apart radios to work out how they worked. And when we went on long walks as a child, he would explain to me how the pistons of a car work. So I've really learnt from my dad to be curious, to try to understand how things work. And absolutely, and this is all the way through my life and from him, is not to be scared of a challenge or a problem. It can always be solved. My mum is the youngest of 7 siblings, mostly girls, 6 girls and a boy, many of whom are teachers, and my mum is a teacher. And I learnt from my mum, well, I'm hoping to learn from my mum, with the importance of communicating, of teaching, and connecting with people. So this is me. I don't know, I'd get that. My name means pomegranate. So Ramona. And if you cut open a pomegranate, you can see it's meant to be full of seed. It is full of seeds, hundreds of seeds. If you don't want to cut it open, you can. I didn't do this, but you can put it in an MRI scanner, and you can see the seeds another way. And that's meant to be auspicious. The seeds are meant to be the good deeds that you could do in your life. So I'm the oldest of 3 girls, and this is my younger sister Natalie, who's watching online. This is, I think, the last time that I was taller than her. This is going to be a bit of a theme. She's incredibly wise, my sister Natalie, and she's an artist and a graphic designer. And here's the 3 of us. In the middle is my sister Leonora. She's incredible. She's really clever, and she's a consultant in public health and works for UK HSA. You can't tell that from here, but maybe you'll get to talk to her later. So what led me to neurology? I mean, Oliver Sacks, for sure, and Sherlock Holmes. So I loved Sherlock Holmes growing up, and I think it was the combination of the detail in the puzzle, solving that puzzle, to be able to see the big picture. And I didn't know it then, but actually there's a very well-established link between Sherlock Holmes and neurology. But I had other influences as a teenager growing up in the 1980s and 90s. Definitely Michael J. Fox, Robert Sean Leonard, for those who remember, and Robin Williams, Alan Alda, Billy Connolly. But actually, turns out I have a connection with all of these people that I didn't know when I was growing up. So Michael J. Fox in the 1990s was diagnosed with Parkinson's disease. Robin Williams, in life, was diagnosed with Parkinson's disease, but after he died in 2014, he was found to have dementia with Lewy bodies, and I've spent a lot of my time in the last few years working with that disease, and his wife has become an amazing advocate of dementia with Lewy bodies. So I got to go to Downing College, Cambridge. I still never really quite understood how that happened. And I had incredible mentors and supervisors. So Barry Everett was Director of Studies and taught me neuroscience, and Trevor Robbins taught me psychology. And well, the reason I chose Downing was it was close to the train station, and there was a large number of medics, and we had fantastic Downing medics. So Danny Gale and Dave Hunt, who are here, which is amazing. And David, when I went back 30 years later, is still much taller than me. And Julia, who I might even be here as well, Julia's a psychiatrist in Cambridge. Fantastic, yes. And then just down the road in Pembroke, I think, was Geraint, who was doing natural sciences and now does medical physics. And you'll hear a bit more about Geraint in a minute. So I went on to do medicine, my clinical studies at UCL Medical School, and I got to do a summer project with Jeremy Rees, and that led to my very first paper. I think you never forget your first paper. I think I remember the page numbers of it, looking at paraneoplastic syndrome. And then together with Jeremy Rees and Rolf Jaeger, I did an imaging project where we were looking at imaging enhancement in patients who had gliomas. And I learned a really important lesson from that project that actually has become important to all the work I do now, which is that if you look at changes at baseline, that some of what's really important is to work out which of those changes are predictive of important clinical outcomes over time. So that was a really important lesson that I learned from that sort of very early project that I did with Jeremy and with Rolf Jaeger. So I did my clinical training at the Middlesex Hospital, at the Hammersmith and the Brompton and the Whittington, and with a brilliant set of SHOs, some of whom I'm still working with, and I met and married Johnny, who actually without Johnny, I don't think I'd have been able to do any of the things that I'm going to be telling you about. So thank you, Johnny, for everything and supporting me and really everything. So that's Johnny. And then for my neurology training, I came here to Queen Square and had incredible teachers and sort of in the different firms. But particularly, I got to meet two mentors. So Andrew Lee's, I was in the movement disorders firm as an SHO, and Nick Fox. I don't know if you remember, Nick, that I was... Where's Nick? There. I was your SHO in the cognitive disorders firm. And I've been lucky to be able to carry on working with many of my mentors, and particularly with Andrew and with Nick. I also had incredible registrars, many of whom are now consultants here at Queen Square, many of whom I work with. So, and the registrars very much taught me a lot of the neurology that I still do now. So then I decided I wanted to do a PhD, and I was very, very lucky that Geraint let me do a PhD in his lab. And funded by the MRC. And Geraint had an incredible lab with some absolutely brilliant people working with, well, Geraint is brilliant, and the people in his lab were particularly brilliant and clever and very nice. And what the lab were working on were different types of optical illusions. So you might think, okay, optical illusions. Well, it was human consciousness, visual consciousness, and I think that was what made me want to go and work in the lab to start with. But why look at optical illusions? So optical illusions really are a clue to how the brain's working. And what each of these people were doing were taking a different example where you could show someone something on a screen, and what appeared on the screen would often stay the same, but the experience of it, or your perception, would differ. And that is the basis for a perfectly designed functional MRI experiment, because you can put someone in the scanner, show something on a screen, and then when they experience something different, you then have different conditions, and you can then look, well, what's happening in that person's brain while they're in the scanner? So what I learned in Geraint's lab was how to test visual function in a quantitative way, and how to design how to carry out and deliver a really, really good scientific experiment. I learned a range of imaging techniques, so magnetoencephalography, MEG, structural and functional MRI. I learned about this relationship between bottom-up information from the brain and top-down information, and that's going to be really important in the work. It is important in the work I do now. And then, really importantly, I learned that it's all very well to do a good, a nice scientific experiment, but you have to be able to explain it properly. And I really learned that in Geraint's lab. So this was the beginning of where I was really learning how to see things differently. So Geraint set me on a project looking at artificial scotomas. I will just say, I never got to present this anywhere because it was so niche. I never gave a talk on my PhD, so you're going to get to hear about it. So what's an artificial scotoma? Well, it's a type of filling in. And what we would do is we would show volunteers a screen covered in black and white, sort of visual noise like snow on a TV. And the person would fixate centrally at a red cross. And in the corner of the screen, the left-hand corner, there would be a black square. And when that person would keep looking at the screen, that black square would seem to disappear. Now, that won't happen for you now because I haven't set it up and I haven't got the bits moving. But what it would look like is this. And what was powerful about this paradigm was that the screen stayed the same, but the perception of it, the experience of it differed. So before I kind of show you what we found, why look at artificial scotomas? And actually, they're really important because our brain we are constantly filling in visual information. So, for example, if you're driving down a country road, you might think that your brain is getting this beautiful view of the lane and the trees coming in through your eyes. But actually, this is more like the information that your brain is getting. Because at the back of the eye, we've got retinal vessels. They're getting in the way of everything. And actually, what the brain is having to do is fill in that information across those retinal vessels to make sense of the world outside. So this was the beginning of actually starting to understand some of the top-down influences of what we see. So I was trying to find a neural signature of filling in. So for the first experiment, I used MEG, magnetoencephalography, and this is me during my PhD in the MEG scanner demonstrating what it looks like. It's perfectly comfortable. I've used this for a lot of patients now, just to show you can smile in the MEG. And what we did was we showed our volunteers this screen with an artificial scotoma, but so that we could keep track of it, we flickered it at 15 hertz frequency. And that meant that we could tell within the MEG, we knew that we were tracking that scotoma, that black rectangle, even when it seemed to disappear. And what we found was that we then could have three conditions. So when we actually made it disappear, so sometimes it really wasn't there. Sometimes it was there and they could see it. And then the third condition was when it seemed to disappear, when they couldn't see it, but it was still on the screen. And we could track that using MEG. And we could also, I then showed in a similar way, I did a similar experiment, and I could sort of replicate the same finding that we could follow that signal using functional MRI. So that was my very first publication from my PhD, my first output really from my PhD, which was really nice. But I had another really important output almost exactly to the day, which was Elia. Almost exactly the same day that paper came out, Elia was born. So she was born at UCH, and she was kept in hospital for a few days afterwards. So the very first time that we got to leave the hospital and go anywhere, I think, actually, Geraint and Mike, you might be happy, we went to the UCL quad, because where else would you go if you were close to UCL? So Elia is a UCL baby. She's just done her A-levels, so time really does go by. So I was interested in sort of this top-down information about what we see. But I got interested in thinking of, like, knowing what we know, knowing what we've seen. And I got talking to Steve Fleming, actually just outside, you would have passed the square just outside. We were sitting in the square having lunch, and we were talking about metacognition, which is knowing what you know. And Steve was doing a lot of work on knowing what you know. And we had an idea that we thought we would work on together. So what is metacognition? Well, it's actually something we do all the time. Every day we're making decisions about the things that we see. So I'll give you a slightly less everyday example. Imagine you're lost at sea, and your only hope of getting back home is a boat coming to save you, coming to rescue you. And you've got a flare, but you've only got one left. So you've got to save that flare for when there really is a boat coming. You see something in the horizon, you're not quite sure. Is that a ship come to rescue me? Now, if it is, you've got to release the flare. But if it's not, if it's just your eyes playing tricks on you, you mustn't release the flare, because then you'll have wasted it, and when a real ship comes along, you won't have it. So that decision is metacognition. It's thinking, how sure am I about what I've just seen? So we decided that we would test metacognition in some volunteers, and also perhaps look at which parts of the brain were involved in metacognition. But when you test metacognition in the lab, you don't use ships, you use a much more dry experiment. So we used screens of stripy circles, which are called Gabors, and we showed our volunteers two screens of 6 stripy circles, and on one of the screens, one of the stripy circles was paler. So we gave them a visual task first. Which slide had the paler circle? Was it slide 1 or slide 2? And then we gave them the metacognition task. How sure are you about this? And they had to make a judgment on a scale, how sure they were. Now, what we did was we varied how difficult we made the task. So when people were really, really good at it, we made it much harder. And when people were really bad at the task, we made it much easier. And what that meant was that everyone doing the visual task was actually performing at the same level. They were all at 70%. And what we found was that even though everyone was at the same level of performance at the visual task, there was a lot of variation in how good they were at knowing what they were seeing. So the metacognition varied quite a lot, and you can see that in the red diamonds. Men and women, by the way, didn't differ, which was quite nice, actually. I think we were quite pleased about that. Now, we'd also asked our volunteers to have a brain scan, and what we found was that that there was a part of the brain where in those people who were better at this metacognitive task, it was bigger. And in those those people who were worse at the metacognitive task, it was smaller, not taller. I've got tall on my brain. So we managed to find a region in the frontal lobe that related to how good people were at metacognition. So meanwhile, I had another baby, Adina. She was born not that long after Elia, and this is Elia meeting her younger sister, also at UCH. And Adina's just finished her GCSEs, so Well done, Adina. I don't know, well done, we'll find out. Anyway. Anyway, I had a toddler and a baby, and I went off to the Royal Free to do my neurology training and got to work with some brilliant neurologists, Lionel Ginsberg. I mean, I really was very lucky to work with Lionel Ginsberg and Jeremy Gibbs, and met some neurologists who I've carried on working with, so Hugh Morris, Tony Shapira, and Annetta Schrag, and some brilliant registrars, especially Joe Swanton and Laura Zambriano, who are my job share partners. And then I had another baby. This is the last one, though. So Hanna, who was born on my birthday, and yeah, she's 12. She hasn't done her GCSEs yet. So I then went to Queen Square to complete my neurology training, and worked with really some giants in neurology, and John Rohr was my job share at Queen Square, and I found that I particularly enjoyed movement disorders and cognitive disorders. So while I was working on the movement disorders firm, one of my jobs was to admit the patients with Parkinson's disease who were coming in for a procedure. I don't know if the consultants knew this at the time, but I did what was expected. I did a neurological examination. It's what you have to do when you admit people. But I also then did cognitive tests on everybody. And now none of these patients had dementia, so I wasn't expecting them to have dementia. I don't know why I did the cognitive test. And I did this brief cognitive test, and one element of that cognitive test is where we ask people to copy intersecting pentagons. And what I noticed when I went round and did these tests were, first of all, most people did quite well because they didn't have dementia. But if they failed on anything, they failed on copying the intersecting pentagons. Now, I didn't know this at the time, but actually a group in Cambridge, so led by Caroline Williams-Gray, had actually done quite a bit of work and showed that the pentagon copying was quite a sensitive and useful test. But I didn't know that at the time, but noticed, did think, well, this is odd. Why are these patients with Parkinson's, they've got movement problem, why are they failing on this particular bit of this cognitive test? So there are some non-neurologists here. So what is Parkinson's disease? I told you that I admitted all these people with Parkinson's. So it was first described just over 200 years ago by James Parkinson, who was an apothecary working in London. And he wrote an essay called The Shaken Palsy, where he described the people that he had seen walking around London. And in his essay, he wrote, it's an amazing description, it's still really valid today, that what he noticed was that people with Parkinson's had this involuntary tremulous motion and a propensity to bend their trunk forward. Now, we know now that this is caused by a problem deep in the brain called the midbrain, and for the non-neurologists, the midbrain is in that red oval there, deep in the brain. And I'm showing you on the left the midbrain of somebody who doesn't have Parkinson's, and what you should be able to see is there's a nice black area, which is where the dopaminergic neurons are. But on the right, somebody who has died with Parkinson's disease, there's loss of that blackness. And so what that causes is a disorder of movement. People get a tremor, they get stiffness and slowness. But and if you look even closer under the microscope, what you can see is inside some of the cells are these inclusions called Lewy bodies, and they were named after Frederick Lewy, who noticed these inclusions in 1912. Now, James Parkinson, because most of his work was actually just watching, he didn't talk to many of the people that he'd watched. He wrote in his Shaking Palsy essay that in Parkinson's, the senses and the intellects are uninjured. But actually, just about 60 years later, Jean-Martin Charcot noticed that at a given point, the mind becomes cloudy and memory is lost. And actually, what we now know is that if you have Parkinson's disease and you follow people up, around 50% of people will get dementia within 10 years. And when I talk to patients in my clinic, who have Parkinson's disease, that's the one thing that really terrifies them. So it seemed to me that I really needed to get to the bottom of dementia and Parkinson's disease. So to do this, I applied for, there was a new type of UCL fellowship, and they gave it this extremely embarrassing name. It's very Austin Powers. They called it an excellence fellowship, and I didn't want to admit to anyone what it was called. Now, it wasn't designed for clinicians, but when I got it, I said to them, look, I still need to finish my neurology training, so can I do it part-time, carry on doing my clinical training? And what that gave me was an incredible opportunity, because what it meant was that I then spent a year doing a weekly Parkinson's clinic with Paul Jarman, and then the year later I did a weekly Parkinson's clinic with Kath Mummery. I don't know if you remember Kath. And from that I got I'm scared to say it here, but probably the world's best training in movement disorders and cognitive disorders. so thank you, both of you, for giving me that training. So that was half the week I got trained in cognitive and movement disorders. And the other part of the week, I could start to try to understand the visual changes that happen in Parkinson's disease, and started to look into, well, what evidence is there? I noticed this pentagon thing, what's going on with vision. So I started to research, well, what is known about visual changes in Parkinson's? But it also meant that I could collect some pilot data. So the pilot data was all really inspired by an observation, a clinical observation that was made by Hugh Morris. And he mentioned to me that he had heard that people with Parkinson's had trouble reading capture images. Now, they've changed over the years, but at the time, there used to be these distorted letters and numbers. And I was quite intrigued. I had this background from my PhD in visual science, and then I was working in movement disorders. Why would people with movement disorders have a problem reading letters and numbers? So I thought, well, let's test this. But I knew from my PhD that you've got to design a really good experiment, and letters and numbers are really confounded by edges and lines. So instead of using letters and numbers, I did images of cats and dogs, because everybody knows what a cat and a dog looks like. But actually what your brain is doing by separating out cats and dogs is quite a complex task. And what I did with these pictures of cats and dogs was I distorted them by sort of stretching them out. And that meant that as I stretched them more and more and more, I could work out which point people could only just tell if they were seeing a cat or a dog. And I could use that. That was what we call the threshold, of where they could just tell which one they were seeing. And that's what I could compare between my people with Parkinson's and my controls. So I invited, it's just a very small pilot project, 20 people with Parkinson's and 11 controls. None of these people had dementia. And I compared this threshold where they could just about tell the difference. And I found that people with Parkinson's were worse at this task. But what was quite interesting was, actually, if you look, there's one person at least was better than all the controls. But then there were a bunch of people with Parkinson's who were clearly much worse. So I got thinking, well, what happens if we follow up these people over time? So we did that, and then a year later, I retested people's cognitive performance. So here on the horizontal axis is how good they were at the cats and dogs test. That's what the patient started to call it, the cats and dogs test. And then on the vertical axis is what happened to their cognition over time. So if they got worse, it went down. And that's exactly what I found. The people who were worse at the cats and dogs test when I first tested them, when I followed them up a year later, were more likely to do worse on their cognitive score. And we actually did a sort of functional MRI experiment, and I worked with Joel Winston to show actually some of where in the brain that was happening. Now, this was quite a small study. It was just a few patients. The cats and dogs test seemed like a nice idea. But I wanted to look in more patients and look sort of more broadly at vision. So the Wellcome Trust gave me some money to do it. And what I did was I invited 100 people with Parkinson's and 40 controls, and we got them to do a whole bunch of different stuff. So we had our cats and dogs test, but we did a few other different visual tests as well. We did some retinal imaging, and this is Louise, who was my very first postdoc, and she's doing optical coherence tomography on me. So the back of the screen is actually, that's my retina that you can see. We took blood for plasma and genetics, because I had been trained by John Hardy, always collect genetics. And we did a whole bunch of different imaging sequences. And we got people to come at baseline, and then 18 months later, and then 18 months after that. So we had 3-year follow-up in people. So she was then my PhD student. I did not know how lucky I was to have Angelica come into my life. She's incredibly talented, and luckily she's stuck around. She's now working as a postdoc, and she's really very much a star in her own right. I mean, really watch Angelica. She's doing her own things, and she's incredibly talented. So she's done, you're going to see quite a lot of things that she got working on in our group. So what she showed when she started to look at the visual changes was that those patients who were bad at the visual test at baseline, when we looked at 18 months later, were more likely to have mild cognitive impairment, or MCI. And they were also more likely to drop on their cognitive tests. This is just the mini-mental state that I had done on my patients when I'd been going around the ward. But then we had 3-year follow-up data. So this is Naomi Hanna, who's, I actually need to say this for everyone, but everybody in my group is brilliant. So Naomi is, so she's just finishing her PhD with me. She's been looking at the 3-year follow-up data. And in blue are the people, this is a survival curve. So to do well, you want to stay flat, and people who do badly are falling down, a bit like going down a slope. So in blue are the people who were good at the visual task at baseline, and in salmon are the people who were bad at the visual task in baseline. And those people were much more likely, by 3 years to have either developed dementia or to be too frail to come for their follow-up visit or even to have died. So it did seem like in our cohort, in our 100 people with Parkinson's, that visual tests seemed to predict changes over time. But I got talking to Bahadur, sort of early on actually, who had been in Geraint's lab. He was a brilliant postdoc in Geraint's lab. And he said to me, well, you've looked at this cats and dogs test, but why don't you set up a web-based platform, put all these visual tests online, and then you can test lots of visual tests at the same time, and then test loads of people. So just to say, this was ages, this was like about 8 years ago he had that idea, and we're only now just analysing our results. I'm looking at Matt, who's now finally, because it's taken, you'll see why it's taken so long. So, and it's not because Matt is slow, this is... So I got a couple of web developers, Ben Jackson and Tristram Gok, and we set up the Vision in Parkinson's study, and we got some clinical information, we did an online visual acuity test, we even did a sort of tapping test that Alison Noyce let us use, and we put a bunch of different visual tests on our web-based platform. But I'm going to show you the one that the patients liked the most, and that was this horses test. So this was actually a painting that I found that was painted by a man called Stephen Gardner, who lives in America, who gave me permission to use his painting on our website. And you'll probably see very easily that there's 7 horses. Everyone can hopefully see the 7 horses, or you've really got a problem. But he's hidden in the picture 22 horses. So have a little look, see if you can find, there should be 22 horses. You have to look very closely. And what we got the patients to do was to click with their mouse where they could see the horses, and it would kind of pop up, and it would kind of go white where the horse was. But look in the water. Yes, someone's, yeah. Look in the sky. So I'm going to make it easy for you. So in the lightning, you can see some. There's one in the water. There's one even in the coat of the horse. There's one in the trees. Anyway, that was the popular one. So these are some results that are really just coming out now that we're just writing out. So because it was on a website, what we then could do was, Elisa Constantini was my research assistant at the time. She got in touch with 33 Parkinson's clinics all over the UK and said to them, well, do you want to test our, I mean, it was a bit more formal than this, but do you want to test out our web-based test with your Parkinson's patients? So this is a map of where all the different clinics that we use. And that meant that we knew that people taking part did have Parkinson's disease because they were being seen by clinical neurologists around the country. They used our web-based test. We collected blood for genetics, because John Hardy says always collect blood for genetics, and we then followed them up over time. So we had baseline and then yearly tests until going out to 4 years. That's why it took so long. And what Matt has been able to show in this separate cohort of 500 patients with Parkinson's and about 100 controls is the same thing. So the patients, so in blue are the people who were good at the web-based test, vision test at baseline. And this was a much shorter form of the test. So I didn't know it was going to work like this. So if you were in blue, your chance of having cognitive impairment by just about 4 years is above 75%. No, you're 75% likely to be all right at 4 years. Whereas if you do badly at the vision test by about 4 years, you've only got about a 25 or 30% chance. So the vision tests really were predicting what was going to happen to people over time. Okay, so that is vision predicting Parkinson's disease over time. But what's going on in the brain? I'd had this whole PhD using brain imaging. I wanted to use it. Now, usually, if you want to understand neurodegenerative disease, what you'll use is measures of atrophy. That's the kind of the classic way, because actually in most degenerative diseases, what you see is atrophy. But atrophy actually is not very sensitive in Parkinson's. That's been really a kind of a problem. You don't see it very much, and when you do see it, it's not very consistent where it is. And together with James Cole and Rohan Bowe, we did find there is a way that we can use atrophy, but essentially we realised we needed to look in a slightly different way. So we used 2 different techniques using brain imaging to really try to understand the brain changes in our patients with Parkinson's. So the first technique was looking at structural connections. And the reason we wanted to use structural connections was that actually in the animal models of Parkinson's disease, the earliest changes are in the fibres, and they're in the axons. So that's where we need to look in humans. And if you want to do that, you need to usually use a form of imaging called diffusion-weighted imaging. But Angelica said, there's this new technique called pixel-based analysis. It's much more sensitive. So you use your diffusion-based weighted imaging, but we're going to analyse it using pixel-based analysis. And when she did that, we get this very robust and accurate measure of the changes in the fibre connections in the brain of people with Parkinson's. So she was able to show that when she compared our people who were good at the visual task and people who were bad at the visual task, that there was breakdown in these brain connections using this pixel-based analysis method. But remember, we had people coming 3 times over a period of time. So what she was then able to show was, we found some changes at baseline, but there were even more changes after 18 months. And when she looked after 3 years, then there's widespread loss of white matter connections. But that's in the people who were bad at the visual task compared to people who were good at the visual task. If you look at Parkinson's patients as a whole, we didn't find those differences. We also had some plasma. So she then said, well, is there a relationship between the fibre connection, the loss of fibre connection, and a measure in the blood? And using, so we looked at a measure called neurofilament light, NFL, and we found that the greater the loss of fibre connections, the higher this level of this plasma. You don't want it to be high, that's bad news. Higher levels of NFL. So that was one measure, structural connections. Another measure that we used was measuring brain iron. And we did this using a technique called quantitative susceptibility mapping, or QSM. And we could do this because we were working with Karen Schmueli, who's a brilliant physicist, and Julio Acosta Cabronero, and George Thomas, who is another person I'm so lucky to have joined my lab and is now sticking around as a postdoc and doing some incredible work in his own right. So George took our patients with Parkinson's and he used this technique, QSM, and he said, well, let's have a look if there's a relationship between their levels of iron in the brain, because QSM is sensitive to levels of iron. I know, Corinne, that's a very simplified way of saying it, but anyway. And so he said, well, how does this QSM measure relate to some of the important clinical measures that we find in our patients? And he found that it related both to motor scores, movement, and to their cognitive scores. And he then showed that if you looked at baseline, that actually this QSM measure could even predict their future movements severity and their cognitive severity over time. So it's nice to be able to measure things, but actually, what about understanding some fundamentals about what's going wrong in the brain? So George said, well, let's take this another step further and use this QSM technique to understand why are some parts of the brain degenerating in Parkinson's and others aren't. So we did this together with Mina Wrighton, who's a brilliant scientist. Now she's gone to Cambridge. And we made use of an atlas called the Human Allen Brain Atlas. And that atlas is, it's actually 6 people who have died of something else. They didn't have Parkinson's disease, but they donated their brains to research. And 180 regions of their brain were extracted and taken out, mashed up, and the genes extracted from those 180 regions. But before those genes were extracted, the brains were scanned. So we've got gene expression data in 180 regions in the brain in MRI space. So what that meant was that George could take the QSM data that we had in our Parkinson's patients, so we know where in the brain there are higher levels of QSM. Sorry, Corinne, I'm simplifying. And he could relate that to the gene expression data in these 180 regions across the brain. And by relating the gene expression data in health with our QSM data in Parkinson's, he could find, well, in the regions where there's too much iron, which genes are being overexpressed in people who don't have Parkinson's. And what he found was it was genes relating to heavy metal processing and to synaptic transmission and cell signaling. So using this technique in our patients, we could then start to drill down into what's actually might be the vulnerabilities. Why do some parts of the brain go wrong in Parkinson's? Well, we had all these different measures. We've got these different, we had retinal imaging and brain imaging and the visual measures. So what we wanted to do as well was to put this all together into a model. So I worked with Neil Oxtoby, who has a model where you can take all these different measures that are done at one time and work out the order, the sequence of events in which they're happening. So this actually, what you do is you read down for the sequence, and the things at the top are the things that he shows using this model happen first, and then as you go down, those are the things that happen later. And what we were able to show with Neil's model was that actually vision changes do happen early, then the iron changes, QSM, and actually that atrophy is a relatively later event in Parkinson's. And we've gone on actually to look at this in a bit more detail, and Zina Schauer has been looking at, I'm looking for Zina, she's been taken this approach, and actually we can start to look at subtypes of Parkinson's using very large databases, public databases of Parkinson's people who've had brain imaging. So I've been able to do, you could probably be able to tell, I've been able to do a lot of the work that I've been able to do because I'm working with brilliant people. So I've been welcomed into the Dementia Research Centre by fantastic colleagues at the Dementia Research Centre. And I've also part and welcomed into the Movement Disorder Centre. So I have absolutely brilliant colleagues in 2 departments, which really means I can get to do some hopefully, through that, it enables me to do some work that's really interesting. But I left everyone behind for a bit in 2018 as part of my Wellcome Fellowship and travelled to Boston to work with Mike Fox. So he's a neurologist, and he was working on a technique called lesion mapping. And his approach was to try to understand all different symptoms in neurology by looking at the underlying functional brain networks and how they all come together. He had a brilliant, very international lab, and this is us. And what I learnt in Boston was that 3 o'clock, if you work with Italians, 3 o'clock is coffee time. Everything stops for coffee time. So Mike got me working on coordinate mapping of Parkinson's hallucinations. And the way he got me to do that was he said, why don't you find all the studies that have been done where people have compared hallucinators with Parkinson's hallucinators with Parkinson's patients who don't hallucinate. Find all the studies that you can, and in each of the studies, they report where there are points of atrophy in the brain. So I found 9 studies, because actually if you're looking for that head-to-head comparison, there weren't that many, found 9 studies. And I've plotted, if you might have to squint, I've plotted in red the points of atrophy that each study found. But I'm going to make it easier for you, so I'll highlight them with some circles. But what you hopefully can see is that they don't line up at all. So if you compare different studies looking at Parkinson's hallucinators versus non-hallucinators, they don't line up. They're all over the brain in different places, and you can kind of plot all the different studies on one brain and see that there's no rhyme or reason to it, really. But Mike's approach was to say, okay, that's the point of atrophy. But what he then does, is his approach is then to say, well, how does that point of atrophy in each study map onto an underlying functional network? So then for each of the different studies that I had found, I then could identify the functional network that point of atrophy was related to. So all the different studies, they look different. There are different functional networks related to that point of atrophy. But what we found was that, I'm showing you now the green oval, almost every study, there was one exception, the point of atrophy that was identified in that study, or the points of atrophy, were related to a functional network that included the lateral geniculate bilaterally. And this is a sort of an overlap. There's no masking here for the sort of imaging people. What we found was there was 89% overlap bilaterally over the lateral geniculate nucleus. So that was quite interesting. But what really was interesting at the time, not that we got a paper, that was just a nice. But what was really interesting was that Nayang Kim was working in Mike Fox's lab at the same time, and she's a neurologist from Seoul, Korea. So she was looking at hallucinations that have happened nothing to do with Parkinson's. So people who happen to have a brain tumour that's causing a hallucination, or a stroke. And she was collecting all the different case reports that had been reported where people had hallucinations due to other things. And what you can see, she's plotted them in red, and they're not in any consistent place in the brain. She then used the same approach. She took these lesions and she worked out, well, what's the underlying functional network that relates to those strokes or brain tumours? And what she found was that the network overlap from those lesions overlapped in exactly the same place that I had found using Parkinson's hallucinations over the lateral geniculate bilaterally. So here's my results next to her results. So it did seem that, through using this approach, looking at Parkinson's hallucinations, so hallucinations that arise from different methods, either from Parkinson's or from other ways, that actually what's fundamentally important is this functional network over the lateral geniculate nucleus. Meanwhile, my family came out and joined me in Boston. So we had that, which was brilliant. I think we still have good memories of being in Boston. We ate a lot of ice cream in the Italian part of Boston, and we got to go to Fenway Stadium and watch a baseball match. So anyway, Boston was really lovely. So I came home and back to London and wanted to work a bit more on hallucinations and Parkinson's. And the motivating factor behind that is that they're common. And they're linked with poor outcomes. So actually, if you have Parkinson's and you develop hallucinations, you're much more likely to be placed in a nursing home. And they're really challenging to treat as well, because the drugs that we use in the clinic to treat Parkinson's will actually worsen people's hallucinations. So to try to understand hallucinations in Parkinson's, we need to think a little bit about vision. And I'd already learnt in Geraint's lab that vision is this active process. So we have bottom-up information that comes from the eyes, but we also have these top-down processes, and these can be memories or previous knowledge, and we call these priors. They're our visual priors. And it's that combination of bottom-up information and top-down information that provides what we see. So the theory that we wanted to look at was, well, in people with Parkinson's who hallucinate, maybe there's well, I already had shown you, hopefully you now believe me, that there's impaired bottom-up. Visual processing is affected in these patients, so we've got impaired bottom-up. But then we wondered, maybe there's too much top-down information. They're relying too much on their top-down priors, and that's why they're hallucinating. So Angelica decided to test this. In fact, this was the very first experiment in your PhD. So to test this, she used these kinds of images that are called Mooney images. Now, if you've seen this picture before, you have to stay quiet. But if you haven't seen it before, the question that Angelica asked people was, do you see a person in this image? So these things are not usually interactive, but maybe if you see a person, put your hand up. Oh, lots of people can see it. Okay, fine. Fine. So this black and white image was made from a colour image, which is a very cute baby in a bucket. Now, once you saw it, yeah. Once you've seen that cute baby, you can't unsee it. So now it's really obvious. So what Angelica did was she took these black and white images, and some of them had a person in, some of them didn't have a person. And then she showed people the underlying template, and then she showed them the original image. And what we wanted to see was how people's performance improves. Now, we expected everyone to get a bit better, a lot better. But what we wanted to see was whether there'd be even more improvement in the hallucinators, because then they're relying more on their priors. And that is what we found. So in green are the people who didn't hallucinate. They did get better, but in the sort of salmony colour, we got even more of an improvement in the people who hallucinated. So we'd shown this is what I call a behavioural task, but what's going on in the brains of people with Parkinson's who are hallucinating. So if you want to look at directions of connections in the brain, you have to use a really complicated form of analysis. And this is what George did, and he did it with some world leaders in this technique called dynamic causal modelling, Peter Zeidman and Adil Razi. And what he did was he took the brain regions that we expected to see changes. So, and you'll notice we included the lateral geniculate nucleus, but we had some other regions as well. We had some bottom regions, so like primary visual cortex, V1, and the thalamus, and then some higher order regions, which is like prefrontal cortex, PFC. And what he then could do, using this complex technique that uses a kind of maths called Bayesian approach, was he then could map out the connections along the top of the connections from each of these regions, and along the vertical side is the connections to each of these regions. Now, you've got to ignore the diagonal, because that's intrinsic connections, and that's actually not as interesting. But essentially, this is a heat map. And what you should be able to see is that there's decreased, so cold colours mean decreased connectivity, and hot colours mean increased connectivity. And what he showed was that there's, in these Parkinson's patients who hallucinate, compared to those who don't hallucinate, that there's decreased bottom-up in the blue, going, let's say, from a lateral geniculate nucleus to primary visual cortex. But then there's also increased connection from the top down, from prefrontal cortex to V1, to primary visual cortex. And you might say, okay, that's quite nice to show those connections, but does it relate to anything real with our Parkinson's patients? So he then took the combination of changes that he found in our Parkinson's hallucinators, and he related that to a clinical questionnaire on hallucinations and showed a very strong relationship between changes in connectivity and hallucination severity. Now, we'd actually done a lot of work looking at structural connectivity, and I'm just kind of showing you. Angelica has taken really a very mathematical approach to the imaging that we've done in our lab, and showed that there's a subnetwork of reduced connectivity if you look at structural networks, and actually she's even been able to look at some of the dynamic functional changes in networks. But I mentioned Andrew Lees, and what Andrew taught me was to try and think outside the box. So he introduced us to the work of William Burroughs, and this is William Burroughs on the left, with Brian Geisen. He was a bit of a beatnik, I think, in the 1960s, and he's sitting in front of a contraption called the Dream Machine. So a Dream Machine is essentially a piece of cardboard that's rolled up with a light inside and holes in it. And if you sit in front of it and you close your eyes, you hallucinate. So Andrew said to me, I've got this, I've had it made, why don't you test it in your patients with Parkinson's disease? So we did that. We got them to sit. We got ethics for this even. We have to get ethics. We got them to sit in front of it. We got them to close their eyes. Sadly, not everything works, and we didn't manage to stimulate any hallucinations. I think there was a missing ingredient that William Burroughs was using. We didn't have ethics for that. So if I've already hopefully shown you, if you have Parkinson's disease, you follow people up for long enough, some people get dementia and they can get these other symptoms of hallucinations and fluctuations and some sleep changes as well. But there's another condition, dementia with Lewy bodies, that's very similar and differs mainly on the timing. So if they get their dementia first, it's called dementia with Lewy bodies. And they're sort of separated by a 1-year rule of how long the time is between when they get the dementia. And if you look at a person's brain who's died of Parkinson's dementia or dementia with Lewy bodies, the pathologist will tell us that they essentially look very similar. So we decided in our group to try and work out, well, are they similar? So this is, I got different students within our lab. So Rohan Bome, who's a psychiatrist, and Naomi Hanna and Evelina Dobreva were all working on different elements of this. And I always said to Naomi, we're going to see if we can find differences, and we might not, but that's life. But we need to do the experiment and compare them. But actually, we found that they probably are different. So we had our patients with Parkinson's dementia and dementia with Lewy bodies, and we had our plasma markers, and we found that overall the plasma, and this is a new plasma marker called p-tau217, that is raised in both of the conditions, not significantly different between the two. But when she used these very clever techniques that Angelica and George had been using, so the brain connections, she found there were differences between Parkinson's dementia and dementia with Lewy bodies. And when Rohan looked at this QSM measure between Parkinson's dementia and dementia with Lewy bodies, he also found differences. And we weren't really expected to see them. So that's something that was quite exciting, to find a difference when you're not sure you'll find it. Now you might think that I love talking about Parkinson's dementia, because I've gone on about it for all this time. But actually, it's taken me a bit of time to enjoy talking about it, and I found that I wasn't alone, and actually that I found that nobody wants to talk about Parkinson's dementia. Clinicians don't want to talk about it, it's kind of uncomfortable, and patients don't want to talk about it because they're scared. But if we don't talk about it, then patients don't get the right treatment, and actually, really importantly, families who are going through a really hard time, they don't get support. So I decided to work on an engagement project with some brilliant artists at Central St Martin's and with some engagement experts at the Functional Imaging Lab. And we decided to develop some toolkits to try to open up these conversations about Parkinson's dementia. So we wanted to do a pair of toolkits, one for people with Parkinson's and their family, and one for healthcare professionals. So because I was working with artists, we did this as an art project. And this is us at Central St Martin's School of Art. And we invited people with Parkinson's to come along with their family members. And we asked them, what do you associate with the word dementia? And how do you want to find out about dementia? But we said to them, don't tell us the answers. Show us using collage and paint and stickers and stick and glue. So this is us all kind of painting and drawing. And they produced it, and we then did let them talk. So they then did tell us what they thought about all these things. And we took these outputs, these collages that they had made, and we then did some focus groups. So we then talked to the patients with Parkinson's and also involved healthcare professionals from all over the UK. And we developed this pair of resources, one for people with Parkinson's on the right and one for healthcare professionals that have been adopted by Parkinson's UK. And they gave us a prize for it, which was very nice. Nice to win a prize. It was my first prize, I think. I never had one for my work before, so that's quite nice. But then alongside these toolkits, I've mentioned this is a really hard condition. It's really tough on family. And Nick Fox had already set up many years ago an amazing, really a sort of a whole, I mean, it's more than a facility, a whole approach called the Rare Dementia Support Service for different types of dementia, for people and their families who have different types of dementia, which is often not recognised because it's different from the more common dementia of Alzheimer's. So Nick very kindly said to me, well, why don't you set up a Lewy body dementia group as part of our Rare Dementia Support Group? And I've been able to do that particularly because of working with the team at the Rare Dementia Support, especially Nikki Zimmerman. And so what we do through that is we have twice a year, we have support group meetings, and also Nikki and her team provide support for our patients with Parkinson's dementia and dementia with Lewy bodies. We're also doing some trials for symptomatic relief. So this is Top Hats, which is a trial for Parkinson's hallucinations, led by Suzanne Rees, and Cobalt, which is a combination treatment adding in memantine for people with Parkinson's dementia and dementia with Lewy bodies, led by John Paul Taylor. Thank you for coming, who is a brilliant psychiatrist. I've learned a lot. I mean, everything really I know about dementia with Lewy bodies I've learned from you, John Paul. So we're testing, so John Paul's leading it, and then we're a site here, testing whether adding in memantine will help. And we're able to do that because of working with the Leonard Wolfson Experimental Centre, particularly with Matt Appleby, who's been extremely helpful and wise. So with John Paul, we realised that actually if we want to improve the lives of patients with dementia with Lewy bodies, we need to get together, we need to work together, and we need to bring together people around the UK who are working in this field. So we set up Lewy Body UK as an annual meeting for researchers and clinicians in Lewy body dementia, and this is us last year in June in Newcastle. And on the back of that, we then set up a Lewy body dementia doctoral training network, where we've brought together 6 centres around the UK and got some funding from the Alzheimer's Society so that we now have a programme and a network, really you called it an institution without walls, so that we can train 25 students with 5 students per year for 5 years who are all going to do PhDs in Lewy body dementia. So it's really, I mean, really to be able to do that with sort of incredible colleagues around the UK is incredible. So I've told you about visual changes and some imaging and dementia with Lewy bodies. So what am I doing now? Well, so far it's one thing to be able to look at the MRI changes, but what I really want to be able to understand is what's the protein changes that's happening in people with Parkinson's dementia and dementia with Lewy bodies. And through the Wellcome Trust, we've now set up a much bigger project where we're getting patients to come along and have loads of scans so that we can really measure everything that we can to do with really the proteins that are building up. So amyloid and tau PET, different types of MRI scans, so 3 Tesla and 7 Tesla MRI scans. We're collecting blood, of course, genetics, but also now doing spinal fluid collection so that we can really get a picture of what are the protein changes underlying this condition. What we want to do is be able to link the changes on MRI with what happens in the brain of people who have Parkinson's dementia. And we've just started a new project that we're kind of building on, where every patient who comes to our project, I ask them if they will donate their brain to the brain bank here at UCL. And so far we've had 2 people who've donated their brain, and this is we've actually scanned their brain, and the brain then goes through neuropathology so that we can link the changes in those patients' brains with the MRI scan of that person's brain, and then even link it back to the MRI in life. But I want to be able to bring these changes also into the clinic, and I've been able to do that thanks to Tarek Yusri, who's really spearheading a project to bring some of these very advanced imaging techniques into clinical radiological pipelines so that we can really bring them to patients. And so really the ultimate aim is I want to be able to use this sort of approach so we can target the right treatment to the right patient and ultimately slow or even prevent dementia and Parkinson's. So I've been able to do this because I have the most brilliant team, and I just thank you. They're brilliant, they're clever, they're also really nice, and I just think you can't ask for much more than that. And I have amazing, wonderful collaborators here at UCL and really across the UK, and some even not in the UK. And also I really can only do this because of my home team, incredible support from home, and my lovely family, my parents, my sisters, and my in-laws. So thank you very much.
Wow, what an amazing lecture. I've known Rimona for 2 decades. And in fact, she was reminding me, I've actually known her husband for even longer. Because he, of course, looked after my daughter when she was in the neonatal intensive care unit at Queen Charlotte's Hospital. So in a strange and slightly weird and creepy kind of way, just as Johnny is really important in Ramona's life, he's quite important in my life too. But enough of that. It's a pleasure and a privilege to offer some remarks about Rimona and her lecture. I love these kind of events because on the one hand, they're the most amazing celebration of academic and clinical achievement that is marked by promotion to professor at UCL. And as Mike said, that is not an easy task, that is a hard task. The bar is high, and both of our colleagues tonight are clearly so far over that bar. that it's amazing. But it's also, apart from being a public celebration, it's also a chance to get a little bit of insight into the personalities of these colleagues. Now, I've known Rimona for 20 years, so I know that lecture was pretty typical. You got a pretty good sense of the individual she is. But I just want to pick out 3 qualities. And I think, no, it's not going to be that embarrassing. It's really nice. That's fine, so always play it straight. Excellence, right? It goes without saying, to be promoted to professor, what you have to do is excellent. But if you look at the excellence that Rimona displays, first of all, it's over a very long period of time. It was a very long time ago that she started being excellent. Now, I'm not going to date it right back to the 70s when she was born. That was a little bit early. I think that's her parents who take the credit for that. But her scientific and academic excellence is over a very long period of time now. Second, it's over a very wide range of activities. Just think about what we've seen. Not just basic science in the laboratory, not just clinical science, but also patient groups winning that prize, doing all of that work, all of those different areas, and excellent in all of them. And it's a particular kind of excellence as well, because you have that sense when you hear that work, oh, I wish I'd thought of that. You know, the clinical observation that sparks a program of inquiry. And I think that's characteristic of much of the work that takes place at the Institute of Neurology and that clinical translational work. Now, the second quality I want to bring out is my favourite word, indefatigability. Right? It's the ability to persist, to keep going. It's the drive, it's the tenacity, it's the slightly annoying terrier-like behaviours as well that you can sometimes exhibit whilst making things happen. And if you sort of pick out moments there when that indefatigability might have been shown, I mean, I got a view of it during her PhD training when, of course, she interpolated 2 wonderful children as well as extraordinarily high-quality papers as well in a miraculously short space of time. But it's also the indefatigability of job share and keeping that academic training going alongside that clinical training. That indefatigability of adapting the Excellence Fellowship, that she hates the word, even though it defines her achievements, at the Excellence Fellowship where she persuaded people to modify it and change it and get a training like this and do that. And dare I say it, in Queen Square, it's even the indefatigability of being part of both movement disorders and the Dementia Research Centre and the slightly complex politics that sometimes characterise that. Gloss over here. So indefatigability. And then the final trait that I'd like to pick out is perhaps the most important one, a generosity of spirit. I think we see that in the way she talked, not just about her family, but the way she talked about you in the room, those who have had the privilege, like I have had, to have play a small part in her development, but also the generosity of spirit in paying it forward to the next generation, and the way she talked about members of her laboratory and current collaborators. So those 3 things I pick out. Why do I do that? Well, obviously it's really nice, and Rimona's a wonderful person. But like all the professors here, and like all the people who are fated to become professors here, you're joining a community. No man is an island, no woman is an island. We work together as a team. And those qualities, I think, are important for that teamwork that characterises our community at UCL. We were founded 199 years ago, as you know. Spoiler alert, we've got a 200th birthday next year. And in contrast to the 2 very important universities in the outer suburbs of London, we were founded because of a commitment not to their polite learning, which continues to this day, but a commitment to useful knowledge. And I think it's those characteristics coupled with what we've heard tonight, and I would include Rob in this as well, because I think he displayed many of the same qualities and spirits, that's commitment to useful knowledge that changes the lives of patients and people around the world, that characterises what we're so proud of at UCL and why we're so proud of Rimona and count her as a valued colleague. So I'm going to hand back to Mike. But before we do so, I'm just going to invite you for one more round of applause for Rimona.
So thank you to both speakers for fantastic lectures. Thank you to Geraint for that really nice vote of thanks. So you're now welcome to join us for a drink in the reception. Thanks very much.